Hi, and welcome back to Chapter 3. We're going to cover proteins in this lecture. So, proteins are one of the most important structure in living things. If you were to dehydrate a tomato, about 50% or more of that dehydrated tomato is going to be proteins. And I know that might sound surprising because you don't think of tomatoes as having a lot of proteins, but they do. 50% or more is going to be how much protein is there. Proteins have tons of different functions. I'm going to go through each of these with you and just give you a brief overview of them. And we might spend some more time on each of these as the semester progresses and as this lecture progresses. So the first function is enzymes, and enzymes are catalysts. They make chemical reactions speed up. The second is defense. We'll talk a little bit about how antibodies form in your body and how they interact with invaders like viruses. The third one is the storage protein. Storage, anytime you have storage, anytime you have storage, you're storing it for later energy use. So egg whites have lots of protein in them, and that's going to be broken down by the developing embryo and used to build up that embryo. So the egg whites themselves are not the embryo. They're basically food for the embryo, or food for you if you're on a high protein diet. Proteins can be used in transport, moving materials in and out of the cells, for example, or even moving things around inside cells. Proteins can be used for cellular communication. And when we talk about cellular communication, we can be talking about hormones. We can be talking about how hormones are released into the blood. For example, insulin is a protein-based hormone. We talked earlier about steroid-based hormones, and hormones are just communication molecules. They can also be involved in nerve signaling, so there might be receptor proteins and signaling proteins that are involved in cell-cell communication. The next one is movement, and when you think about protein, you often think about muscles, and that's good. But movement can also be how single cells move, like a sperm has a flagella, and that helps it move around in space. Some cells have cilia, and that helps things move around as well. Our last one is support, structural support, and for structural support, here we're talking about things like collagen or keratin, and these are really important in your skin, or, for example, keratin is in your hair. Insects and spiders use structural proteins to build cocoons or webs and things along those lines. So, proteins are really important, and probably one of the most important ones, though, is enzymes. And I don't think it's unfair to say that without enzymes, we could not have life. Enzymes make chemical reactions happen faster. And we'll be learning a lot about this eventually, but just as a kind of a preview here for you, enzymes bond to substrates, and the enzyme substrate complex causes the chemical reaction to happen in the substrate. Now you have something called the product. And remember, those are terms that we've used before. But notice that the enzyme stays exactly the same. So enzymes aren't used up as when they're catalysts. They just cause the substrate to change. Proteins are made up of polypeptides, and polypeptides are made up of amino acids, and there are 20 different kinds of amino acids that are found in nature. Put them together in any one of thousands and thousands of ways, and you bond each of these amino acids together, you form a chain. That chain is called the polypeptide. it's not called a protein until after the polypeptide folds. And so we're going to be talking about protein folding, and that's going to be a very important part of understanding what proteins are. But let's go back and start talking about the amino acids. Now, you've seen this before. We've talked about this when we were talking about functional groups. An amino acid is a very specific molecule that has a carboxyl group, also known as an acid group, and an amino group attached to a central carbon. The central carbon also has a hydrogen coming off of it, and really importantly, has a side chain. And the side chain has the name R for if it's not very specific. So the R group or the side chain does have specific versions depending on which of the 20 amino acids you're talking about. Let's take a look at some of those amino acids. These are categorized by how they interact with other, basically the nature of their side chains. So the R groups are highlighted in yellow here, and they're on the top of the amino acid. Here it was showing you as on the bottom, but it doesn't really matter which position it looks like it's in. The R group is the one that's variable. And notice that the amino acid group is down here. There's the acid, there's the amino group, there's the central carbon and the hydrogen. Those are always the same. It's the R group that varies. This is just a list of non-polar side chains. No, I am not going to make you memorize these. I'm just showing you that sometimes those side chains or the R groups can be hydrophobic, and I do want you to know that. Sometimes the side chains can be hydrophilic, like you see here, and sometimes they can be electrically charged. And if it's electrically charged, of course it's hydrophilic, but in this case it forms that ion. So notice that there are 20 of them, and they each have a different set of functional groups, and those are important. Now, amino acids are going to be linked together in the same way that all polymers are linked together, and that is through a dehydration reaction. But in the case of polypeptides, the name of the bond that forms between amino acids is called a peptide bond, and that's literally why we call it a polypeptide. We don't call them polyamino acids, we call them polypeptides, because the name of the bond that forms between this amino group and this acid group is called a peptide bond. Do you remember the name of the bonds that attach between fatty acids and the glycerol molecule? Or how about the name of the bond that attaches for the carbohydrates? If not, you might want to go back and review. So, polypeptides. Polypeptides can range in length from fairly short, just a few amino acids connected together, to really, really big. We're talking about thousands and thousands of monomers connected together. Each polypeptide has a unique linear sequence of amino acids. So here we see a linear sequence of amino acids. And notice that the carboxyl end is attached to the amino end. We call that the C terminus connects to the N terminus in our peptide bond. If a protein is going to be functional, it needs to have a very specific shape. And a functional protein is basically, we're going to be looking at how it twists and folds and coils up into that unique shape in order to do the function that it has to have. So if we look at here, if we look at this protein that goes through a membrane, the regions in the center are going to have a very specific shape. The region on the ends are going to have a specific shape. Same thing for enzymes. Depending on the shape of the enzyme will depend on what function it has. And so we are going to learn a little bit about shape. It's the amino acid sequence that leads to the three-dimensional shape. So the amino acid sequence is important as well as the shape. And again, a protein's shape or structure tells you what it's going to do. Shape is so important when it comes to proteins. This is a great example of it. Here what you see is a complementary shape between an antibody. Remember we talked about how that's involved in the immune system just a couple slides ago. And this is a protein from a flu virus. And notice how they have a very similar shape. They're going to be interacting with each other in this region right here. If they had different shapes, then the antibody would not be able to bond to the protein from the flu virus and wouldn't be able to take it out. So that shape is extremely important in proteins. When we think about shape of proteins, we think about shape in 4 different levels, which are called structures. So the first 3 of those structures are important in single polypeptides. So this is a single polypeptide, and we can talk about the primary structure, secondary structure, and tertiary structure of this polypeptide. This is the primary structure, which is the sequence of amino acids. We'll talk a bit about the secondary structure, which can be either in a pleated sheet or an alpha helix. We can talk about the tertiary structures, which is how it folds into this little ball of protein. And then we can actually also talk about the quaternary structure, which we see that word right here, quaternary structure. And the quaternary structure happens when 2 separate amino acid polypeptide chains interact with each other. So let's hit these 4 levels of protein structure in a little bit more detail. The primary structure, again, is a unique sequence of amino acids. Going back here, a unique sequence of amino acids, each of those amino acids having different side chains. Yeah. So the unique sequence of amino acids is the primary structure. The secondary structure is found in most proteins is either coils or folds in the polypeptide chain. It's a result of hydrogen bonds between, the amino acids that are fairly close by to each other. And the secondary structures include either this thing called the alpha helix, which really does look like a slinky, or it could be a beta pleated sheet. So this fold right here is caused by several hydrogen bonds between fairly close by amino acids. You have to think about this a little misleading here. This is a single polypeptide that's being folded back and forth into this beta pleated sheet. That's all because of hydrogen bonds. So we've got this linear structure right here, right, in the primary structure. And then the secondary structure is hydrogen bonds kind of stabilizing the polypeptide backbone into the helix or the pleated sheet. Then we have the tertiary structure. And the tertiary structure is involved when you have regions that are fairly far away from each other, interacting with either van der Waals or disulfide bridges or ionic bonds. And let's take a little bit closer look at that. So here's our tertiary structure. Again, really, really important with the interaction between side chains, and there's different kinds of interactions that might happen. With the secondary structure, it's all about hydrogen bonds, right, which we see here. With the tertiary structure, it can be hydrophobic actions like van der Waals. Remember, hydrophobic are those nonpolar interactions. It can be disulfide bridges. So example, 2 cysteine amino acids. Again, we don't memorize that, but cysteine has sulfur in it. So a disulfide bridge can happen between 2 cysteine molecules. Or it can be ionic bonds. And again, go back and take a look at those polar amino acids that I showed you on the previous slides. The quaternary structure is really simple. This is a polypeptide, this is a polypeptide. When those two polypeptides join together, you have the quaternary structure. You can have quaternary structure with 4 polypeptides joining together, or more. Quaternary structure. An example of why it's so important to understand primary structure is looking at diseases, genetic diseases. And a good example that we'll be talking about throughout the semester is sickle cell disease. You've probably heard of it. Sickle cell disease is inherited, so it's genetic, and it is a disorder of the blood. This blood disorder causes red blood cells to kind of clog up the blood vessels. And what happens is you have just one single tiny little change from one amino acid to another. That substitution changes the folding of the hemoglobin, and that folded hemoglobin causes the red blood cells to have this sickle shape, like that. So those sickle shapes get stuck in the tiny little blood vessels and clog them up. So they kind of like do this jamming thing, and they jam up there. Whereas the round red blood cells just kind of slip through, like so. These kind of jam into each other and cause blood, and that causes a sickle cell crisis. People die if they're in a sickle cell crisis, or they can die if they don't get treatment quickly enough. So again, showing you a little bit better version of that. There's the sickle shape, and again, this is only because of a single change in this amino acid right here. Up here, it was GLU. It changed to VAL, which changes the secondary and tertiary structures, which causes, when there's low oxygen around or stress around, to form these very tight aggregations, and that's what causes the change in the shape of the blood cell. There are lots of things that can affect protein structure besides changing the amino acid. For example, the physical conditions that you have, or the chemical conditions that you're having in your body, can affect what that protein structure looks like. If you change the pH in your body, if you change the salt concentration of your body, or the temperature. For example, if you have a fever, your body temperature rises, and you can cause the proteins to basically unravel. Here they are in this beautiful tertiary structure, but if there's too much heat, or if the pH is wrong, or if there's too much salt, the protein can become denatured, and when it doesn't have the right shape, it doesn't have its function. So that means it becomes biologically inactive. Not a good thing. I'm not trying to scare you with the temperature. Your body has fever, and your fever is there to try to denature the invader, but sometimes it also ends up denaturing your own body's proteins, and that can lead to death. All right, well, we're moving on to the 5th part, and that is all about nucleic acids. See you in a minute.